What determines the operator's specific interaction with core or repressor? Complementary structures between proteins and DNA. That's gonna be determine this issue. Previous session I explained the determining the things regarding the operator. The second half will be how complementary structures could be formed. So many things are actually involved in this process. The binding of sequence-specific and non-specific binding, as I mentioned, in the red region and middle region with the green color. And also positioning of the recognition helix correctly in major group. Lastly, altogether could lead to what? Complementary surface formed between the proteins and DNA over nearly all protein-DNA contact regions. Not only to the proteins, but also to the DNA. Very interesting aspect about the protein-DNA specific interactions. Local conformational changes in the DNA rather than direct sequence-specific DNA-protein interactions modulate repressor and core binding to operators. The remaining things will be about the allosteric effect. We already had some concept about allosteric effect, so we may skip this one this time. Trip repressor is involved in the triboperon, by which the an amino acid tryptophan could be biosynthesized in E. coli. This is a trip repressor, alpha protein. Fourth and fifth make helix-turn helix. It actually works as dimeric form. The 2 and 1 and 2 helix they just make core regions, and then 4 and 5 together with 6 making head regions. 3 helices just connecting two regions. In this case, T3 repressor itself just remain inactive because two heads containing recognition helix simply doesn't fit to the major group, or two major groups apart 34 angstrom. As you see, the structures. But when tryptophan, called core repressor, binds to P3 repressor can cause conformational change in a way to recognition helix fit very well to the major group, about 10 base pairs apart. It turns out co-repressor tryptophan just binds to the between the helix 5 and 3, just changing their structures. Second type will be lock repressor. This protein has 3 domains actually. Amino terminal DNA binding domain in red with the helix-turn helix motif, and large core domain having 2 subdomains, green and blue, where the co-repressor lactose binds to. Lastly, C-terminal alpha helix contributing to make tetramer. The functional unit is tetramer. The C-terminal alpha helix make 4 helix bundle. So each 2 dimeric forms make 2 alpha helix-turn helix. So each just could bind to the major group of target DNA, which has palindromic the sequences. Pro R is a regulator for the purine biosynthesis operon. Structure actually behave assembled into dimer, but the behave actually sequences are very similar to lock repressor and working similarly. So N-terminal having helix-turn helix being inserted into major group of binding site, and then small helix being bound to the minor group between them. Chapt stands for the catabolized gene activating proteins. Actually, in the presence of cyclic AMP, it changed into active dimer, so that it could bind to the specific region of DNA shown here. Actually, it binds to large lengths of DNA, 27 to 30. Actually, it behaves as dimer with different colors. But you see the recognition helix in red color being inserted into major group. And then the interesting feature in this DNA Chapt cyclic AMP shown in this purple color has some unique feature. The central 10 base pair forms B-type DNA, and then the following sequence are very highly conserved with T, G, and this region has a 4-degree kink, very sharp the turn, turning changing directions, and then forming regular B-forms on both sides. So almost break up substance structure in these regions. Each polypeptide unit has 2 domains. Basically 12 beta strands and many alpha helices. The colored region of the upper part represents Helixton helix, and the lower domain has a binding site for the cyclic AMP in pink color. So in general, in prokaryotes, positive regulations could be either induction and repressor. Separation activator itself could be inactive, but when it binds to inducer, it could be active, or it could be opposite. Itself is in active state, but with the repressor, it could be inactive. For the negative regulation is the opposite case. For induction, repressor is active alone, but in the presence of inducer, it could be inactive. Repressor, repression, the repressor is inactive, but together with the co-repressor, it could be